So good afternoon, everyone, and good evening. It's really a pleasure to welcome you for a new Cancer Cachexia seminar. And today it's a special day for us because we will have one of our trainees talk session. And so we will have the chance to listen to three young speakers today, Dr. Arneson-Wissink, Dr. Angelino, and Dr. Chovsepian. I have a few announcements to make just before leaving the floor to the scientists. So first, I'd like to advertise the next seminar. The next seminar will be on July 12th, and we will have the chance to host Professor Andreas Fischer. He's an expert in endothelium metabolism and adipose tissue, and his talk will be about endothelium and how endothelium is important to mediate adipose tissue remodeling in cancer. So the hypothesis is that the tumor release factor and those factors will interact with the blood vessel wall. This will induce a response in the endothelium, and this remodeling will lead to a remodeling of the adipose tissue. And so he will be presenting data on that topic. And so we invite you to join us. I think it will be a very interesting and interactive session. The second announcement is that I guess most of you know, but we will have soon the conference of our society. The conference of the society is organized every 2 years, and for the first time it will take place in Europe. So it will be really a nice occasion for Cancer Cachexia people to meet, exchange, initiate collaboration. And so we encourage everybody to apply for a poster and to join us. As the member of the educational committee, we are organizing several events, and I'd like to take a chance today to advertise them a bit. So the first thing we are organizing is a networking event. So it's really open to everyone. Everyone can apply. It really will be an opportunity for networking, for discussing. If you have a Cachexia superhero you want to meet, just put them in the comment and we'll do everything to put you in contact if you want to have mentoring advice. So you just need to register, and I'll show you in a bit where you can register. So you register and we will come back to you before the conference and give you more detail. The second option for networking that we have set up is called a bit of a speed networking. So it works a bit like the speed dating. We will define the list of topic or science icebreaker that you can discuss with the person, and then every defined time you have to change seats and start discussing with another person. And so we plan to have maximum between 30 and 40 people that will be participating to that. And for that event, again, we ask you to register so that we can organize it the best way. So I put the link in the chat. But what I wanted to show you, it's very easy to find. So you just go in the program. So you go on our website, you go here in conference, annual conference and program, and then you will see those two events there, and you have the form. So here's the form for the mentoring event, and you can click on them and you can reserve your spot here. And so then after that, we come back to you and we'll get it organized. So that said, that was for all the announcement. And so it's my time now to introduce the first speaker of today. So the first speaker of today is Paige, is Dr. Paige Arneson-Wissink. She completed a PhD at the Mayo Clinic in 2021 under the mentorship of Dr. Jason Dawes. And today she will be presenting work from her postdoctoral work in the lab of Dr. Aaron Grossberg and Dr. Dan Marks at Oregon Health and Science University. I'll stop sharing my screen so that Paige, you can take the floor. The title of our talk is Hepatic STAT3 signaling drives muscle loss in Cancer Cachexia. So Paige, thank you for being with us, and we are listening to you.
Thank you so much for that introduction. Can everyone hear me all right? Okay. All right, so like she said, today I'm going to be discussing the role of the liver in contributing to cancer cachexia. Oops. And since I think we're all in this wonderful forum pretty familiar with the general basics of cancer cachexia, I'm going to kind of jump right into the content today. And I want to encourage everyone to sort of consider the nutritional paradox that exists in cachexia, where on one hand, we know that undernutrition is a critical component of this syndrome, and we have several factors that are contributing to that, including decreased appetite, hypercatabolism in peripheral tissue, and in pancreatic cancer, impaired exocrine function. But on the other hand, we also know that nutritional support is not sufficient to prevent or reverse cancer cachexia. And this made us think about what metabolic deficits could exist in a cachectic patient that are making them vulnerable to this state of chronic undernutrition and sort of preventing traditional nutritional supplementation from being an effective therapeutic. And in order to answer that question, we need to consider the adaptive response to undernutrition, which is a metabolic response that occurs in healthy individuals and is led by the liver. And the main function of this metabolic adaptation is to provide energy substrates, particularly to the brain. And the brain can really only use glucose and this other type of molecule called the ketone body in order to function properly. And the liver is really great at generating these substrates, and it actually has 3 different mechanisms by which glucose and ketones can be generated when the intake of sugar is low. And so the liver uses these 3 different substrates: glycogen, which is actually stored and released from the liver; fatty acids, which are liberated from the catabolism of adipose tissue; and amino acids, which can be liberated from the catabolism of skeletal muscle tissue. And we know from starvation studies that there's actually a preferential use of these substrates where fatty acids are favored through the pathway of ketogenesis, which generates ketone bodies, to be used prior to amino acids are used derived from the skeletal muscle. And in this manner, it's hypothesized that metabolism of lipids from adipose stores is actually helping to preserve muscle mass in cases of prolonged undernutrition. But what's really interesting about ketogenesis in particular is that we don't just use it in cases of prolonged starvation, but it's used in acute events of fasting as well, including in humans in particular in overnight fasting during the sleep cycle. And so this is a metabolic process that's employed on a daily basis. And it's also important to consider that in cachexia, it seems that this sort of this sequential use of substrates appears to be disrupted, because we know that adipose tissue and muscle tissue catabolism occur concurrently in the case of cancer cachexia. And that led us to question whether lipid metabolism and in the liver, this adaptive response could be reprogrammed in cancer cachexia. So with all of that in mind, we proposed 3 study goals, which were to identify the metabolic changes associated with early stage cachexia, investigate the connection between inflammatory signaling and disrupted metabolism, and to determine whether disrupted hepatic metabolism is driving cancer cachexia. And we have this model framework we're working off of where we know undernutrition should induce lipid oxidation and ketogenesis in the liver, and that this provides some benefit to muscle and resilience to an individual during periods of low nutrition. But we're working with a model of pancreatic ductal adenocarcinoma, and we're asking whether factors released from the tumor are signaling in the liver and what the metabolic response to this event is, and whether this is driving cachectic phenotypes. So we've hypothesized that pancreatic ductal adenocarcinoma drives an impaired hepatic adaptive response to undernutrition, which is exacerbating the systemic energy deficit and resulting in skeletal muscle wasting. And to address this hypothesis, we're using a model of PDAC in mice where pancreatic cancer cells, KPC cells, are implanted orthotopically into syngeneic animals. And we started by looking at a model for early stage PDAC cachexia, and we were interested in an early time point because we wanted to be measuring metabolic effects that were potentially driving the onset of cachexia rather than metabolic effects that are the output of increased sickness in the animals. So we're looking at 7 days post orthotopic implantation of pancreatic tumors. And you can see that at this time point, tumors have developed in the pancreas. And when we do a body composition analysis, we don't see any significant differences at this time point between fat mass and lean mass in sham and PDAC tumor-bearing animals. And this was also true when we looked at gross muscle mass. So then we implemented a food restriction where mice were restricted to 50% of their baseline food intake. And we did this to mimic the undernutritive anorectic state that's experienced by cachectic patients, and to put stress on this adaptive metabolic response that should be occurring when animals are fasted. So we can see that food restriction did not influence tumor development. And when we looked at fat mass, we see, as expected, both sham and PDAC animals have lost fat mass in response to the restricted caloric intake. However, when we looked at lean mass and gross gastrocnemius muscle mass, we see a preferential loss in lean tissue only during food restriction in the PDAC animals, indicating that there is increased susceptibility for muscle loss during food restriction in PDAC. And this was accompanied by a significant increase in the expression of TRIM63, which is a catabolic gene expressed in the muscle. So it appears as though PDAC mice that are food restricted have this increased propensity for muscle catabolism. And so we were, of course, interested in whether that was correlated with changes in adaptive lipid metabolism. And to measure that, we first looked at gene expression of mitochondrial lipid import genes, beta oxidation genes, and ketogenic genes in the liver of sham and PDAC animals. And we saw a decrease in the expression of all of these genes really in the PDAC mice compared to the sham animals, indicating that there's quite heavy disruption of this genetic program, at least in these mice. So then we next tested this functionally using an octanoate challenge, where we inject mice intraperitoneally with the medium chain fatty acid octanoate after a fast, and then measure circulating ketone levels for 3 hours. We can see that sham mice maintain their blood ketone levels equivalent to their fasted state, but PDAC mice are incapable of doing this. So together, this transcriptional data and the functional data both show that there's an impaired response to fasting in the PDAC mice. And in particular, this is manifesting as impaired ketogenesis. Because we know that inflammatory signaling is really critically linked with cachexia onset and progression, we next questioned whether inflammatory signaling was also causing this impaired ketogenic response in the PDAC mice. So I'm sure everyone on this call knows that IL-6 has been very heavily implicated as a driver of PDAC cachexia. And so we used a model where we could assess the effects of IL-6 on hepatic metabolism. And we used a whole body knockout IL-6 mouse compared to wild type controls. And it's important to note that the particular KPC cell line we use doesn't produce IL-6 itself. So when these cells are implanted into the knockout animals, we don't detect any circulating IL-6 in the PDAC mice. But this knockout model does not have an effect on pancreatic tumor development. So we see equivalent pancreatic tumor mass. However, when we look at gastric mass, we see preservation of muscle tissue in the IL-6 knockout mice, which is paired with no upregulation of catabolic gene programs in the muscle. So when we looked in the same mice at the lipid metabolism, we first again looked at gene expression of lipid metabolism genes and ketogenesis related genes. And we see an increase in expression of these genes in the IL-6 knockout PDAC mice relative to the control PDAC mice, suggesting that blocking IL-6 signaling provides a benefit to ketogenic signaling in the liver. And functionally, in an octanoate challenge, we can also see here that the IL-6 knockout PDAC mice are maintaining blood ketone levels after octanoate injection much more efficiently than the wild type PDAC animals are. To assess whether IL-6 really is sufficient to prevent ketogenesis in hepatocytes, we then used an in vitro cell model where we cultured AML12 hepatocytes in the presence of octanoate, which induces ketogenesis. You can see that here. and IL-6. So we found that IL-6 is significantly upregulating phosphorylation of STAT3 protein, indicating that this inflammatory signaling pathway is activated in the hepatocytes. And we can see that while the octanoate treatment alone increases total ketones released into the cell supernatant, the addition of IL-6 blunts this ketone release, accompanied by a significant decrease in HMGCS2 expression. HMGCS2 is the rate limiting step of ketogenesis. So these data together show that in this sort of paired in vitro and in vivo set of experiments, that IL-6 is sufficient to prevent ketogenesis in hepatocytes. And since we've observed that ketogenesis, impaired ketogenesis is linked with early stage PDAC, we finally asked if blocking inflammatory signaling at the level of the liver would prevent cachectic phenotypes in the muscle. And to address this question, we used a liver-specific knockout of STAT3 so that we were blocking IL-6 inflammatory signaling only on the level of the hepatocytes. And these were compared to littermate control animals. We can see that both genotypes did not have a difference in pancreatic tumor mass, and there was no difference in circulating IL-6 levels. When we looked at gene expression of beta oxidation in ketogenic genes, we again saw that the liver-specific loss of STAT3 increased expression of both beta oxidation and ketogenic genes relative to the control PDAC. And when we looked functionally, we also saw an increase in circulating blood ketone levels in the liver STAT3 knockout PDACs compared to control PDAC mice. And most impressively from this study, we found that the liver-specific loss of STAT3 was sufficient to preserve muscle mass in PDAC mice at levels equivalent to the sham animals, while the controls lost significant muscle mass. So to conclude, we've shown that early stage cachexia is characterized by impaired liver lipid metabolism and ketogenesis. That IL-6 is sufficient to impair ketogenesis in the liver, and that hepatic STAT3 knockout prevents ketogenic and muscle deficits in PDAC mice. So we return to our model. Undernutrition states should increase beta oxidation, lipid oxidation, and ketogenesis in the liver, but we've shown that IL-6 released from the tumor is blocking this via signaling through STAT3, which results in reduced ketone production by the liver. And we believe that reduced ketone production by the liver is sufficient to lead to muscle wasting and the onset of cachexia in PDAC mice. So I'll just wrap up with a big thank you to both the Grossberg and Marks Labs for their support in this work, especially Heike and Jessica, who directly contributed to this work, and our funding sources and the audience for your attention. And I am happy to take any questions. Thank you.
Thank you very much, Paige, for this very exciting talk. I guess there's a lot of questions. So feel free to ask questions either in the chat, or if you want to just switch on your microphone and switch on your video, feel free to interact with us. Selma Masri, you want to ask a question, please, Selma?
Hi, Paige. Can you hear me?
Yes, I can.
Perfect. Great talk. That was awesome. I have two questions for you, both kind of quick. Have you, I know the KPC model is a bit difficult to work with, but have you gone into a GEM model to see if you can recapitulate some of these results there? Just because of the 7-day timing is super fast. And the second question is, where do you think is the source of IL-6? You were mentioning a little bit about tumor derived, but in PDAC, a lot of that major source of inflammation is really stromally derived, especially cancer-associated fibroblasts. And so, what are your thoughts on that? I'm just curious.
These are both great questions. Thank you so much. So your first question was about using the KPC GEM model. So we have done some work in the KPC GEM model, and we've seen the recapitulation of the sort of impaired fasting ketone levels in the KPC mice. We haven't pursued work further with those animals, but both in that model and in some patient clinical data, we see that PDAC individuals have impaired fasting ketone levels. And your second question about where the IL-6 is coming from is also a fabulous question. We asked the same question because we do see elevated IL-6 levels in the PDAC mice, but the cells, the KPC cells themselves don't seem to produce IL-6. We did some in-depth flow cytometry analysis of the tumor, and it seems like the most IL-6 producing populations were cancer-associated fibroblasts and also immune cells present in the tumor. Yeah.
Awesome. Great talk.
Thank you.
Stefan, you want to take the next question?
Yes, thank you. And congrats on some amazing data, Paige. I was wondering a couple of weeks ago, there was a study in Nature suggesting there's a crosstalk between the primary tumor and the liver via extracellular vesicles that then affects the Kupffer cells in the liver. And I was wondering whether you looked into measured anything with extracellular vesicles or the Kupffer cells or stellate cells within the liver. So it's basically the liver microenvironment and how.
We haven't assessed extracellular vesicles in our model. Since we are able to detect the free IL-6, that was sort of the target of our studies. But I think that certainly there's a complex milieu of things released from the tumor and cytokines contained in the vesicles or other components of the vesicles could be interesting too. And we haven't done an in-depth analysis of the liver microenvironment either, but I think that could be a really interesting work. We've had some discussions about the role of intrahepatic immune infiltration and the effects that could have on this model as well. Thank you.
Thank you.
Due to time constraint, I suggest we go with one last question with Anisha, and then maybe Paige, you can answer the other question through the chat.
Yeah, absolutely.
Thank you.
Hi, Paige. Wonderful data and brilliant talk. So I just had a curious question that in your mice with the ad lib and the nutrition restriction, did you check the inflammatory signatures of the plasma?
So we've measured IL-6 in those animals. We didn't do like a cytokine array to look at more
so
broad.
The IL-6 levels are the same with nutrition restriction.
Was blunted slightly with the nutritional restriction. I think blunted slightly with the nutritional restriction, but
okay.
But the same trend higher than the sham animals. Yeah.
Okay.
Yeah.
How long was the food restriction?
We did food restriction for 3 days. So the final 3 days prior to endpoint. Yeah.
Thank you so much.
Thank you, everyone.
Thank you again for sharing. I suggest, Elia, maybe you can start sharing your screen. So it's my pleasure to introduce Dr. Elia Angelino. So Dr. Angelino got his PhD in medical biotechnology in 2017 at the University of Eastern Piedmont in Novara in the laboratory of Andrea Grazzini. There he studied the role of ghrelin in contracting muscle atrophy and in promoting muscle regeneration. From 2017 to 2020, he worked at the San Raffaele University in Milan, Italy. And now he's currently working in the lab of Andrea Grazzini at the University of Turin and at the University of Eastern Piedmont. And there he's studying molecular mechanisms underlying cachexia and sarcopenia. And today we'll be discussing his presentation, Restoring Cyclic AMP Signaling Counteracts Mitochondrial Dysfunction and Muscle Wasting During Cancer Cachexia. So Elia, thank you for being here, and we are listening to you.
Thank you for the kind introduction, and thanks for also to the committee for this opportunity. So I can skip this introduction. So this is a graph that shows a volcano plot of our RNA-seq analysis that we made in the bile gastrocnemius muscle at 13 days post C26 injection. And this is quite well known that there is a huge modulation at transcriptional level of 1,000 of gene, and you can see that there is a quite symmetric distribution between the downregulated and the upregulated genes. And also, if we performed an enrichment analysis, we can clearly see, as expected, that the upregulated genes are enriched in the pro-inflammatory signaling and like STAT3 dependent or NF-kappa B dependent, and on RNA processing and ubiquitin mediated catabolic process. However, what I want to highlight is that the strongest enrichment is among the downregulated genes, and in particular, the top 6 terms that came out from this enrichment are all terms that are related to mitochondrial function, in particular to the electron transport chain, subunit, and ATP synthesis, and other mitochondrial-related. So little is known about the mechanism that underline the upstream mechanism that underline this downregulation that is so coordinated and so synchronized toward the oxidative metabolism. And when we performed the enrichment analysis for transcription factor loss of function, we clearly saw a strong enrichment in the cyclic AMP responsive element binding protein, CREB, transcription factor loss of function, downregulation. So this suggests that genes, downregulated genes, some of the several downregulated genes in the muscle of cachectic mice could be CREB direct or indirect target genes. And these include also several ATC, electron transport chain subunit encoding genes. So why this can be important? Because CREB is, as you know, the transcriptional effector of the cyclic AMP signaling, that it binds on the CMP responsive element within the promoter of the genes. And the signaling is sustained in muscle by the tonic release of norepinephrine or noradrenaline that from the sympathetic nerve. The norepinephrine activates the beta-2 adrenergic receptor, and that is a G-alpha-S coupled receptor that induce the production of cyclic AMP and activation of pKa, that in turn is a kinase that has several substrates, and CREB is one of the most important substrates that is phosphorylated and thus activated by pKa. In this signaling, another important player are the phosphodiesterases that hydrolyze the cyclic AMP to AMP, and so are negative regulator of the signaling. These axes, the sympathetic and adrenergic cyclic AMP and CREB axis, regulate under physiological conditions several aspects of the muscle, such as we maintain the muscle mass, the oxidative metabolism, and the neuromuscular junction integrity by acting at the post-synaptic level in particular, and overall maintaining the function of the tissue. So interestingly, we observed that some key players of the signaling, of the cyclic AMP signaling, are differentially expressed in the muscle of cachectic mice. In particular, the phosphodiesterases 4 isoform are upregulated, so the negative regulator of the signaling, and while catalytic and regulatory subunit of the pKa are downregulated, and also the G-alpha-S protein expression, gene expression is downregulated. So we basically asked whether cancer cachexia, the cachectic environment, procachectic environment, could reduce the cyclic AMP or sympathetic cyclic AMP axis, and thus contributing to the phenotype that is observed during cancer cachexia. And first of all, we check whether the sympathetic nerve activity is modulated during cachexia within the muscle, and this is not the case. You need the measurement of norepinephrine content in the skeletal muscle. That is a proxy of sympathetic activity, does not change after following C26 injection in the muscle. And so we try to understand whether there is whether the procachectic environment could induce a cell-autonomous defective cyclic AMP signaling. And to do so, we generate TCM from two different models, so C26 and the Lewis-Lang carcinoma. Then we treat the C2, C12 for 1 day, and then we challenge these cells with isoproterenol, that is an agonist of the beta-2 adrenergic receptor, and we evaluate the cyclic AMP response. And so through a threat-based sensor of cyclic AMP, you can see that the treatment with both the TCM is able to strongly reduce and impair the production of cyclic AMP. Here you can see the dynamic of the production through time, and this is the total quantification. Also, the downstream signaling is impaired upon TCM treatment. This is CRAB phosphorylation PK dependent, and also at transcriptional level, CRAB in the presence of the TCM is less active. And these are this is the Cree luciferase reporter, and this is the expression of the endogenous PGC1 alpha and NOR1, that are genes that contain the Cree site and are target of the cyclic AMP signal. So overall, we can say in vitro in this system that the TCM impair the cyclic AMP axis, in particular acting, since that the action is upstream, so in the cyclic AMP production. And as I said before, PDE are the enzyme that hydrolyze the cyclic AMP. In particular, PDE4 are the predominant isoform that hydrolyze the cyclic AMP in the muscle. And there are 4 different members, the A, B, C, and D. And basically, we observe that the PDE4B and D are upregulated upon treatment with C26 TCM or LC TCM, confirming what we already observed in vivo in the C26 mice. So we just try to inhibit with a pan inhibitor of the PDE4 isoform, that is Rolipram, and see what happened. And as you can see, when we add the Rolipram, we saw a complete rescue of the cyclic AMP production, and these are the total quantification upon TCM treatment. And to try to dissect whether there is one of the 2 isoform, PD4B and 4D, could play a major role in this effect, we generate knockout cells for PD4B and 4D. And we saw that basically the PD4B knockout, here the total quantification of this graph, behave like the wild type cells, so they are sensitive to the reduction of cyclic AMP induced by TCM. However, as you can see, the PD4D knockout cells increase so much the production of cyclic AMP, both in control condition and also in the C26 TCM treatment. And moreover, when we add the Rolipram, so the PDE4 pan inhibitor, you can see that the PDE4B knockout and the wild type, as expected for the wild type, they immediately rise up the level of cyclic AMP. However, the PDE4D knockout cells, so the green line, are completely insensitive to Rolipram. This could indicate that the PDE4D isoform is the isoform that restrains the cyclic AMP in the C2, C12 cells. So we use Rolipram in vivo. We treated mice with Rolipram in the C26 experiment. And first of all, we look at, as you can see, the tumor size weight and the weight loss and the epidermal fat are not changed by the treatment with Rolipram. And also the cytokine expression, RNA expression in the tumor bulk is not changed by Rolipram treatment, in particular of IL-1 beta, IL-6, and TNF-alpha. The muscle, we observe that muscle is slightly protected against atrophy Indeed, the Rolipram slightly increased the mass of the muscle, gastrocnemius and quadriceps, and accordingly to this, it reduced the expression of the atrogenes, the MORF1 and atrogen1. And also the cross-sectional area show that Rolipram treatment slightly protect against atrophy, the type, in particular type 2X and type 2B fiber that, as you know, are the most sensitive to atrophy in the cancer associated cachexia system. Also, we take a look on the postsynaptic NMJ structure, and as you can see, in particular on the volume of the postsynaptic compartment through bungarotoxin staining of the acetylcholine receptor. And we observed that the Rolipram treatment rescue the decrease of the volume of the postsynaptic NMJ. And this, and also it seems to protect against the decline of the force measured through the grid test. However, we are interested also in the transcriptional changes, and what we observe is that Rolipram, so PDE4 inhibition, modulate the expression of a subset of genes, in particular the 23% of the genes that are modulated in cachectic muscle. However, when we look at the hierarchical clustering of these genes, we clearly saw that the effect of Rolipram is to rescue the expression toward the control. So the green is the Rolipram group. You can see that, for example, these subset of genes are downregulated in cachectic mice, and upon Rolipram treatment, these genes are again upregulated, and vice versa for the downregulated one. So for the upregulated one. Looking at what these genes are enriched for, we clearly see a very strong enrichment here in the genes that are downregulated during cachexia and together upregulated by Rolipram. And these these genes are part of are enriched in term related to mitochondrial function and the mitochondrial electrotransport chain, and also mitochondrial biogenesis. So this is just to show that the engineering pathway analysis that several subunits of the complex of the electrotransport chain and of the ATP synthase are indeed upregulated upon Rolipram treatment and are regulated during cachexia. Looking at the muscle and the mitochondria in the muscle, we observed that the PDE4 targeting rescue the loss of mitochondrial mass within the muscle. We saw this through mitochondrial copy number quantification, and also it's clearly visible through the Western blot with VUDAC that is we use as housekeeping of mitochondria. Finally, we also observed that at the functional level, the oxygen consumption rate is rescued in the skeletal muscle. This is an Ouroboros experiment that we made in collaboration with Simone Reano and Nicoletta Filigetto from Novara from the explanted muscle. You can see that the overall respiration is strongly decreased in the C26 mice, vehicle treated. Rolipram treatment rescue, partially or completely rescue this decline. So in conclusion, I can say that for this in vivo part, that PDE4 targeting in cachectic mice restore the expression of mitochondria and oxidative metabolism related genes, and restore the mitochondrial mass in muscle and the overall tissue oxidative function. And also ameliorate the postsynaptic structure of the NMJ and the grid strength. So my statement is that the defective cyclic AMP signaling and sympathetic sensitivity within the muscle could contribute to muscle wasting during cancer cachexia. I would like to thank all the lab, in particular Andrea Graziani, that is the PI of the lab, and Lorenza Bodo, that is the PhD student that did several of these experiments. and also Valeria Malacarne, Shua Barua, and Fabiana, and all the other people. And more importantly, also our collaborator, in particular, Roberta Sartori from University of Padova, and Marco Sandri, and also Giuseppe Caretti, and several other people, and you for the attention.
Thank you very much, Elia, right on time. So there's already one question in the chat. So the question is, what about the status of adrenergic receptor on cachectic muscle?
The adrenergic receptor in cachectic muscle, we don't know in vivo, is at the transcriptional level, there are no changes in the expression. For what concern the protein expression, we try to detect the receptor, but we didn't was able to do, probably because they are, as other GPCR, they are very less expressed. And what we know is that in the cell, in the context in which we treat with the TCM, we know that the expression, both the total expression and the surface exposure of the receptor is comparable following TCM treatment.
Thank you. Saba, please, you want to ask your question?
Yes, can you hear me?
Yeah.
Yeah, Elia, that was really a very nice talk and quite informative. My question is about your last slide. If you normalize your respiration per mitochondrial content, do you see actually a change in mitochondrial respiration?
Thanks. We did not normalize over the mitochondrial content. I don't know if it's possible to do by normalizing toward I don't know, copy number or something else.
Or VDAC, or VDAC.
Or VDAC, okay. So
because you are changing mitochondrial content.
Yeah.
I just want to know whether the changes that you see is actually reflected as intrinsic changes in the mitochondria or simply changes in mitochondrial content.
Yes, sure. I can do. I will do. I think that so our impression is that is everything the function. So the change in the function of the mitochondria, the OCR, are related to mitochondrial content. So are a direct consequence of the mitochondrial content. Indeed, also when we look, you can see this slide still. Yeah. When we look at the expression at protein level of the subunit of the complexes, we saw like this only if we lyse the muscle. But if we try also to isolate the mitochondria, and so by normalizing the number of the mitochondria, we didn't observe any reduction even in the C26 muscle. So it seems that a matter of number of mitochondria, and mitochondria probably biogenesis and also maybe mitophagy that has been shown to be very important. Thanks for this.
That's what I was thinking of. Yeah. Thanks. Thank you.
Thanks to you.
That's really informative. Thank you very much. For sake of time, I think we stop here. But thank you again for your presentation, and it was really, really nice to listen to you. Now I invite the third and last speaker of today to take the floor and share a screen. It's Dr. Alexandra Chovsepian. Dr. Alexandra Chovsepian got a master's degree in cognitive neuroscience at the Radboud University in the Netherlands. And then she got a PhD in neurobiology at the University of Munich in 2014. She did a postdoc, and now she's a postdoctoral scientist at the Helmholtz Center of Munich. And the title of our talk today is Diabetes Promotes Cancer Cachexia, a Deadly Combination. So Alexandra, thank you for being with us, and we are listening to you.
So hello from my side. Can everyone hear me?
Yes, we do, and we see your slide.
Perfect. Just a moment. Yes. Thank you very much for your introduction and for the opportunity to present today. So yes, as said, my story today is about how diabetes promotes cancer cachexia and why this is a deadly combination. Okay, yes, it is moving now. So Just a moment. Okay, I'm not seeing the upper part of my screen anyway. So first of all, a little bit about diabetes and what is diabetes and how it develops. So in healthy individuals, after a meal, glucose is released in the blood, and as a response, we have insulin secreted by the pancreas. And I want you to imagine insulin working as a gatekeeper when it binds to its receptor on cells in the periphery, and thereby opens the door for the glucose to enter the cell. So what happens in diabetes is that we have excessive levels of glucose in the blood, partially also due to prolonged Western diet, for example. And then we have excessive insulin secretion. Insulin continuously binds to its receptor until the receptor gets desensitized, and cells fail to respond to insulin properly, which we call insulin resistance. And also, after this, glucose cannot efficiently enter the cell. And after a while, a pancreas is trying to compensate for this by producing more and more insulin, and until failure, basically, so until the pancreatic beta cells cannot produce any more insulin. And therefore, we have increased levels of glucose in the blood. And for some reason, okay, now it works, sorry. And for this blood glucose frenzy has grave consequences. And diabetes creates basically a domino effect of complications with, for example, developing cardiovascular disease, stroke, diabetic dementia. We have problems with the eyes, non-alcoholic fatty liver disease, problems with the kidneys, also diabetic neuropathy, neuropathic pain, as well as foot ulceration, which can lead to amputation. Although a lot of people suffer from diabetes, we don't hear a lot of people dying directly as a cause from diabetes. However, diabetes is a silent killer, and it has many ways to kill. Some of those is by inducing a heart attack or a stroke, kidney failure, diabetic ketoacidosis, hyperosmolar or hyperglycemic state, and infections or sepsis. And yet another way in which diabetes can kill is by increasing the chances and the risk of cancer. And this is a recent study. It's a review of 25 studies that have found that diabetes increases the risk of several cancers, as you can see here. And this is not surprising, as we know that many tumors rely their energy production on glycolysis, and therefore they can really thrive in the hyperglycemic environment of diabetes. And yet we have found recently another way in which diabetes can lead to mortality, which is by increasing the risk of cancer cachexia. And this is exactly what our recent paper was about, published in Molecular Metabolism, with title Diabetes Increases Mortality in Patients with Pancreatic and Colorectal Cancer by Promoting Cachexia and its Associated Inflammatory Status. And we were very happy to hear that it will be on the cover of July's issue. Now, I also want to talk a lot about the physiology of cancer cachexia, as most of us are acquainted, but I will highlight the fact that cancer cachexia is a multi-organ metabolic syndrome that has many similarities with type 2 diabetes, including insulin resistance and altered glucose homeostasis. So a little bit about the design of our study. We started with a large database of 605 patients, This was provided by our collaborator from the Technical University of Munich. Then we identified the largest cancer groups, which were pancreatic and colorectal cancer. And those patients had also known diabetes status based on previous medical history. We combined those groups and having a cohort of 345 patients. And as a control group, we used 62 patients without any tumor or any inflammatory diagnosis. And then we analyzed those patients in terms of their BMI and weight loss, in terms of their survival, in terms of body composition via CT scan, including adipose tissue and muscle mass. Of course, we looked at several known cachexia parameters, and we also did a retrospective validation for plasma glucose and insulin. And now to our findings. We found that in cancer patients with pancreatic or colorectal cancer, basically 26% of them were diabetic, while only 15% of them were obese. Diabetes really reduced the probability of survival in cancer patients, while obesity didn't have this effect. And diabetes also increased the weight loss compared to cancer patients without diabetes. And again, we didn't observe something like that for obesity. And then we saw that patients with type 2 diabetes had higher levels of C-reactive protein and interleukin-2 inflammatory markers also associated with cachexia compared to cancer patients without diabetes and control patients. And they had also lower albumin and hemoglobin levels, which are indicative of organ damage, for example, kidney damage or malnutrition in case of albumin. And low hemoglobin is an indication of anemia, which is also very common in cancer cachexia. And as you can see, there was no such effect in obese or overweight individuals. The only effect that we see is that patients with normal weight had higher interleukin, because in this group here, there are some cachectic patients also included. So the main message here is that it's cancer and not obesity that basically reduces the probability of survival and leads to higher weight loss and increased inflammatory factors, and also reduce albumin and hemoglobin. Then we wanted to really validate the diabetes status of our cohort because, as I said, it was based on previous diagnosis. And we basically saw that patients with cancer and diabetes had abnormally high levels of fasted glucose, higher than patients with cancer, and patients with cancer had more or less normal levels, meaning that they were not misdiagnosed or wrongly undiagnosed. And of course, fasted blood glucose levels really correlated with blood glucose levels measured at a random fed state. And these were also all measurements done in the clinic. And then we also validated in the lab that indeed the random plasma glucose levels of diabetic patients were much higher than just cancer patients. Interestingly enough, higher fasted blood glucose correlated with higher weight loss and higher levels of inflammation. And we also looked at glycated hemoglobin, and high glycated hemoglobin also saw the trend for higher weight loss. And this reflects basically the blood sugar levels of about 2 months duration. So we saw that high glucose is indeed a contributor to weight loss and inflammation in those diabetic patients. Of course, we saw that diabetes did not affect the tumor aggression. It's basically shown here that it doesn't alter the grade of the tumor, or it doesn't worsen the staging of the tumor. And although in patients without diabetes, so just cancer patients, we saw that not having a tumor progression increases the chances of survival. However, in patients with diabetes, having no tumor progression did not create such a difference. So it's basically that saying that diabetes patients have a worse outcome anyway, independently on their tumor progression. And then we really wanted to describe very well our cohort and see how different parameters can influence cancer cachexia or diabetes or survival, and also interact with each other. And here we made a correlation matrix for all these parameters. And basically everything that has color is significant. So I know that there's a lot of information here, but I want you to focus on, for example, these factors here, which are all cachexia related factors, and they really related positively with death events. So cancer cachexia correlated with death events, with CRP levels, with being a female, with older age, with higher leukocytes, and of course with having diabetes. Interestingly, cachexia didn't correlate with any of the adipose tissue values. However, it negatively correlated with important proteins such as hemoglobin and albumin and the total protein. And it also negatively correlated with skeletal muscle. And for diabetes, we have a lot of similar interactions. Of course, as we said, it correlated with weight loss and cancer cachexia. It also correlated with being female, with age and leukocytes. Interestingly, it positively correlated with intramuscular fat, and of course, negatively correlated with survival, and as well as hemoglobin and albumin, and again, negative correlation with skeletal muscle. So basically, cachexia and diabetes are correlated and have also many common associated parameters. And then we indeed saw that the incidence of cachexia is higher in those patients with diabetes than in patients without diabetes. So it's about 80 to 60% of them. And it's those patients that have diabetes and cachexia coexistence that have the worst survival outcome, much worse from all other groups. For example, for cancer and diabetes, but without cachexia, or in cancer and cachexia without diabetes. So we can say that the effect of diabetes on survival is mediated by cachexia. On the other hand, diabetes is often associated with higher BMIs, and obesity is also frequently associated with higher weight loss. For example, that's something that happens in COVID. So we wanted to make sure that BMI or fat mass are not do not have any hidden effect in what we see on survival. So they are not covariates of the effect in diabetes and cachexia coexistence on survival. And for that, we first saw that indeed BMI or total adipose tissue area index do not correlate with survival in any of the groups. And after that, we performed an analysis of covariance or ANCOVA. So in that case, the program, yes, exactly, the statistical program, in that case, we used SPSS or Excel. The program basically creates a BMI covariate adjusted model. And we see that the mean of the survival days between the adjusted model and the simple normal model is not significantly different in any of the groups. And the same is true for the adipose tissue. So this means that BMI or TATI are not significant covariates. However, what is significant here and really influences the survival outcome is the group. So having cancer, diabetes, and cachexia. And we wanted to be thorough and also look at other potential covariates in the effect of diabetes and cachexia on survival. And we saw that age, sex, obesity, fasting glucose, or CRP are not significant covariates, and only hemoglobin showed up as a very significant covariate.
Alexandra, sorry to interrupt. Can you wrap up in the minute so that we stay on time?
Yes, sorry.
Thank you.
Yes. So then we looked at other inflammatory markers that predict mortality, and they are all higher in diabetes or diabetes and cachexia, meaning that these are the main, diabetes and cachexia are the main factors in reducing survival. And yes, this is my final slide. We found that specifically in patients with pancreatic cancer, the incidence of diabetes is even higher. Again, we have the same effect on survival, less survival, higher weight loss, higher hospitalization dates in this patients with diabetes and cachexia. Sorry, in patients with diabetes. And we found a very specific signature of patients with diabetes and cachexia coexistence, having really high levels of inflammatory CRP and very low levels of hemoglobin and albumin. And therefore, the take-home message is that cancer and diabetes really reduces survival, increases weight loss, and in those patients that have both, they have the worst outcome in terms of survival, weight loss, and inflammation. And our conclusion is that cachexia may be considered as an emerging diabetes complication. And of course, it highlights the importance of closely monitoring diabetes in cancer patients with cachexia or with risk of cachexia, the need for a timely diagnosis and early medication, and also for maybe nutritional intervention, including sufficient calories, but avoiding foods that would make diabetes worse. And of course, the next step is to identify the mechanistic link between the two, and we will work on that in vivo in the next time. So I would like to thank my institute, our group leader, Maria Rome, and of course, our collaborator from the Technical University of Munich. And with that, I would like to thank you all for being here. And yeah, I'm waiting for questions. Thank you.
Thank you so much, Alexandra. Roberta, please go ahead.
Yeah.
Thank you, Alexandra. I have a couple of questions. The first one is it possible to discriminate diabetes secondary to pancreatic cancer per se?
Yes. So in our case, we did that. I have it as a complementary study. I will share my screen again so you can see. Just one moment. Yes. So, yes, exactly. So, yeah. So what we did is that in this group of pancreatic cancer, we separated between the patients that were diagnosed a long time ago, so had a long-term diabetes, with patients that had the recent diabetes, because this, of course, recent diabetes could be an effect of the pancreatic cancer. And indeed, you can see that the recent diabetes ones had a much lower plasma insulin than the chronic diabetes. And the same goes for HOMA-IR, which reflects insulin resistance. And so we think that those patients are basically, they are having type 3 diabetes. However, this is about a quarter of the pancreatic cancer patients. However, we didn't exclude them from this study because what we saw is that insulin did not really correlate with survival or weight loss. So for us, they are not so different. In terms of glucose, they are not different from the other patients, basically. So we did not exclude them.
And all the diabetic patients were treated or untreated against diabetes? And if yes, did you notice any differences in the type of medications that per se could affect muscles, for example?
Exactly. Let me find, yes, exactly. So it's here. Yeah, the problem is we had only 30 patients with written down medication. So I was also surprised because I was expecting more of them to be treated. I'm wondering if it is a lack of writing it down, or just they were not treated. These are the type of treatments that they got. Most of them, however, had insulin or metformin. But as you can see, there was no difference in weight loss or survival between treated and not treated.
Okay.
Yeah.
Thank you.
Thank you.
Are there other questions? If no, I suggest we close here. I want to thank again the three speakers for their bright talk today. I think it was very fascinating talk and very complimentary in all of them. We will have a second session of trainee talk in the next semester, so stay posted. And we hope to see you all on the Andreas Fischer seminar next in July. So enjoy your day. Bye.